Okay, we're going to talk about the phenomenon of epistasis. This is where the effect of one gene is hidden by the effect of another one. And the example, there are lots of different examples. The one I'm going to use is the coat color in dogs, in labradors. And they can be yellow, black, or brown. So let's see what's happening there. All right, so we start off with, there's a brown pigment in the dog, and that is converted into a black pigment by an enzyme. And let's say that the enzyme is coded for by the enzyme is coded for by gene B. Right, B is a dominant gene. Okay, so if you are that, you can convert the enzyme, can convert the pigment into the black. If you are recessive, the dog can't convert. So the pigment stays brown. Okay, so where does the yellow bit come from then? Well, that is because there is another protein involved here, another gene product, and that is required to incorporate the pigment into the hair. So you've either got your brown or black pigment there. And it needs to be incorporated into the hair. All right, and to do that, it needs needs an enzyme coded for by gene I, we'll call it. And again, that is dominant. So if we have a look here, we just write down here. If you've got either homozygous dominant or heterozygous, they can incorporate. So the dog can be black or brown with that genotype. And then if it's a homozygous recessive, it can't incorporate. So those dogs are always going to be yellow, no matter what the status of the B gene is. So if we make a list of all the possible genotypes you can get from those two different genes, okay, and they are here. Now we can see, right, so we can see that in order for the dog to be yellow, it's got to have that that phenotype. So that's our yellow ones. Then of the other ones remaining, which ones can be brown? Well, it's the one with the double, the homozygous recessive BB there. So you get those two brown, and everything else can incorporate the pigment, and they can convert the brown pigment into the black, so they're all going to be black. So there you've got three phenotypes: yellow, brown, and black. Now the next thing we're going to do is we're going to do a cross between two double heterozygotes. Okay, so let's say we get these dogs. Okay, so two double heterozygotes. And what would they be actually? They would both be black dogs, wouldn't they? Have a black phenotype. We're going to cross them and see what ratio we get. Now, normally when we do this kind of dihybrid cross, we'll get a 9 to 3 to 3 to 1 ratio. But we'll see in this case, we're not going to get that because we've got epistasis, because the effect of the B gene is sometimes masked by the effect of the I gene. So let's do our normal square down here. Right, so on the top line here, we've got the female gametes. Here, and we've got the male gametes here. Right, So let's color code what color we're going to get there. So right, first of all, let's look up for the yellow ones. Now, for any yellow ones, you need a double. You need to have that genotype there. So that's going to be yellow. That's going to be yellow. That's going to be yellow. Now let's look for the brown ones. Now for the brown ones, of the remaining ones, you need to have a double mutation in the B gene. So let's say we've got that one, that one, that one, and that's it, isn't it? Okay, and everything else is going to be black. Okay, so what is the ratio we're going to get then? Well, we've got 4 yellow, we've got 3 brown, and we have got 9 black. So it's not quite the usual 9:3 to 3:1 ratio that we get from these crosses. As I say, it's caused by epistasis, because the genotype in the B gene is sometimes masked if you have this double mutation in the I gene.